giving us this webinar today. I will let Sean introduce you in the lecture. I am Laura Dubé, the founding chair and scientific director of the McGill Center for the Convergence of Health and Economics. I'm also a chair in consumer and lifestyle psychology in the Desautels Faculty of Management. We have started this series about 3 years ago, I believe, called The Bridge series, very much within the context of addressing health challenges that are at the interface of two things, of what we do on the economic as well as on the social side of our everyday life and our everyday society and economy, but also at the interface with the science of understanding how the human brain guide decision making in real world context. There is a lot of synergy between complexity science, multi-scale processors, and so on, that are yet to be explored within the context of the abundance of data we are around. Can we transform those into insights that can help us tweak a bit of our model of modern society and economy in a more adaptive manner? So that's in a nutshell this Bridge series that is co-hosted with the Pittsburgh Supercomputing Center, as well as with the Healthy Brain, Healthy Life initiative here at McGill, and Mission 2.0, which is a complexity science model initiative at NIH. Back to you, Sean.
Great, and so I'll let Jay DePass introduce the PSC component of this. Jay, if you want to unmute and give us a introduction to the PSC.
Okay, great. So Jay DePass, I'm the Director of Public Health Applications at the Pittsburgh Supercomputing Center, which is a joint venture between Carnegie Mellon University and the University of Pittsburgh. And we're participating in the Bridge webinar as part of the MIDAS, Modeling Infectious Disease Agent Study in Mission 2.0. So this is broadening the scope of that infectious disease modeling and allowing us to incorporate a little more realism in the agents that we model, and testing the boundaries of the kinds of modeling that's done in the MIDAS group. So sorry for the little bit of a rambling introduction. I'll turn it back over to Sean and say that I'm very excited to see the work presented here today.
Great, and my name's Sean Brown. I'm the Associate Director of Research Software Development at the McGill Center for Integrative Neuroscience, and I'm representing the Healthy Brains for Healthy Lives project, where I'm the Chief Technology Officer of the NeuroHub platform, which will be a place for bringing together multi-scale, multidisciplinary, and multimodal data with novel forms of analytics to solve the problems that are plaguing the health and well-being of the Canadian population. And it's my pleasure, I get the pleasure of introducing Dr. McIntosh, who will be our presenter today. Since 2010, Dr. McIntosh has served as the Vice President of Research at the Baycrest Center and the Director of and a Senior Scientist at the Rotman Institute, which is also at Baycrest. He is also a professor in the Department of Psychology at the University of Toronto. His research is geared to the development of a unified theory of brain operation that emphasizes the integrative capacity of the brain based on the theory that cognitive operations emerge from the interactions between brain areas rather than being the sole responsibility of a single region. It focuses on two components: technical developments to explore brain integration and collecting experimental evidence of that integration. His areas of study include neurodegenerative diseases, personalized neuroscience, and also, interestingly enough, developing immersive educational entertainment experiences. He helped secure the initial investment to bring together computational, cognitive, and clinical neuroscientists to establish the project that he'll be talking about today, the Virtual Brain, which is a super exciting project. I'm really excited to hear the talk today. He received his PhD in psychology, behavioral neuroscience, and statistics at the University of Texas, Austin in 1992, and then in the less warm climes of the University of Calgary, Alberta. He received his Master's of Science and Bachelor of Science, and now resides where it snows quite a bit more than in Austin. So thank you, Randy, for agreeing to talk to us, and we're really excited about hearing the presentation. So I'll turn it over to you.
Great. Thank you, Sean, and thank you, everybody, for attending today. I'm not sure if it was mentioned, but I'll try and make sure I leave time for questions. If there is something you're missing in terms of hearing me and so on, please note something in the chat window, and then we can loop back later as well to cover anything you may not have understood. In terms of the presentation today, what I'd like to do first is basically do my last slide first, which is the acknowledgement slide, which people tend to leave at the last, and what ends up happening is you sort of miss some stuff. So I just wanted to make sure I had a chance to acknowledge both the people that contributed to the science, and that's over on the left-hand side of the slide. Myself, Victor Yersa in France, and Petra Ritter in Germany. The three of us are basically the main partners that built the platform, but there was a lot of collaborators that contributed different bits and pieces to it. Mike Breakspear in Australia, Gustavo Deco in Barcelona, Steve Small and Anna Solodkin, who were in Irvine, California, recently moved to UT Dallas, and Olaf Sporns in Indiana University. What you'll see also was a technological marvel in the sense of being able to pull together software packages that were written in different languages, putting them all under a unified package, and that's really the heavy work by the team listed in our technology here. People who are in industry, who are working with Victor, working with myself, Tanya in particular has actually been working with me for a number of years, trying to push this thing forward. So just by way of acknowledging people that really contributed to what I'm going to talk about today, it's definitely team science, team science, as my mind, team science at its best, actually. Okay. So by way of what I'm going to try and cover in the next, say, roughly hour, is to, first of all, start with talking about the sort of base that led to the development of the virtual brain, both the theory as well as a little bit into the architecture of the model itself. Then I'll move into several applications. The first talking about this ubiquitous idea of resting state networks in the brain, or the intrinsic activity of the brain, basically what's happening when you're not doing anything else. Your brain's always active, and the question is, can we use that to understand the cognitive capacity of your brain? And then I'll go through several clinical applications. The first one looking at modeling of Alzheimer's, dementia, and stroke, looking at the balance of local and global parameters in the models themselves and its relationship to cognitive status, and also using the model itself to actually predict therapeutic outcomes. And I'll end off with a little bit more detail on the model of epilepsy, which is being led by Victor Yersa in France, telling you how you've been able to use that to actually to help guide, for example, surgical interventions, which I'll talk about in a few minutes. Okay. The idea of scales has been mentioned in the introduction to my presentation, and it is something that's inherent in complex systems, generally speaking. In the brain per se, there are a number of scales over which things operate. You can think about the microscopic scale, where you've got individual neurons and their various synapses, and the synaptic activity themselves are very microscopic, of course. You then move to the sort of mesoscopic scale, where you're talking about the operations that are happening within parts of the brain, between individual neurons, and potentially some of the interactions between different segments. Cortex and thalamus are shown here, for instance. And then you have the macroscopic scale, much of what we deal with in neuroimaging, where you're looking at activity of large pieces of tissue, and how those pieces of tissue basically are interacting when engaged in different kinds of cognitive and behavioral tasks. These scales are ubiquitous in complex systems, and do present a problem in terms of understanding how the system is actually operating. This is a generic slide. This is borrowed from the group in the New England Complex Systems Institute in New England, of all places. And the notion here is that, generally speaking, complex systems have multiple spatial and temporal scales that influence one another. So you see down at the bottom, you may have simple aspects of the system. So for example, individual units, for example, or individual atoms for that, in that case, that then form aggregates and move up the scales, interacting, forming different units that again interact at a different scale, then again at a different scale. And through all these scales that are piled on top of one another, you have emergent behavior that characterizes certain aspects of that system's operations. So modeling complex systems brings an appreciation for these multi-scale influences. And as I mentioned earlier, the brain, in fact, is a very good example of a complex system. It's difficult to model, but actually I think modeling is probably one of the best ways to try and understand some of the main features of these systems. It comes at a cost. There are efforts, as you probably know, for modeling the brain at different levels. There's a long history of modeling individual neurons, a more recent history of trying to model large-scale systems, either at the individual cell level or, like the virtual brain, modeling things at the network level. The challenge, of course, is that complex systems are nonlinear. Complex systems are indeterminate in terms of how you move between different levels. There are many routes that you get to one kind of emergent behavior that are different combinations of subordinate elements below. That's shown in this particular slide here. This is actually borrowed from an excellent review by Yves Marder and Goyard in Nature Review Neuroscience in 2006. I'm not going to go into the details of this particular slide. I do encourage you to go read the review itself. The main point here is that you have a large collection of parameters that result in certain kinds of behaviors. So it's not a one-to-one mapping between one parameter value and a particular emergent behavior. There's actually a number of different ways these levels can interact with one another, and it's actually a combinatorial explosion in some contexts. This means if you're going to model complex systems, you do need to make some decisions of what you're going to ignore in terms of the underlying behavior and what you're going to focus on. And that's part and parcel of why I think we've been successful with the virtual brain. So we focused on the scale of large-scale brain networks and how they enable cognitive function. So let's dive into the platform itself. I'm hoping everybody has coffee or tea to sip. The platform is open source. You can download it. This is the website here. It works in most major operating systems, Windows, Mac, and Linux. It's written in Python with a graphics user interface cover, so you can run it from a web browser. You can also run it from the command line using Python code. There's a number of people who are doing that, so it's quite flexible in that context. You can run it on a laptop, you can run it on desktop, you can run it on a high-performance computing cluster as well. All these scales, of course, will depend on the detail of the model you're trying to run, but the notion there, again, is that it is flexible. You can run it on most platforms without much trouble. In addition to downloading, we do have educational opportunities. We have these things called node workshops. We've had 8 or 7 so far. There's one coming up in Marseille for those of you who are chiming in from Europe. If you're close to France, close to Marseille, there will be a 2-day workshop on May 20 and 21st. You can get more information on the website, on the Virtual Brain website itself, or go to the Eventbrite web link on the bottom of the page here. But these are both hands-on as well as tutorial didactic lectures to give you an idea of dynamical systems theory, but also then being able to create models in real time. So it's worthwhile going if you're in the area. This slide here is giving you an idea of the general workflow for the virtual brain. If we go in this circle here, we start with real data, empirical data, to try and provide the initial framework for the model. So we can start with individual cortical geometry. So for example, taking an MRI, structural MRI, extracting the brain surface and using that to represent the geometry of the brain. Also using MRI, we can estimate the connections between different parts of the brain using, for example, diffuse tensor imaging and estimating the tracks from that connect different parts of the brain. So we have the geometry, we have the structural connectivity matrix. That gives us sort of the structural foundation for the model, and then we can introduce dynamics using what's called the neural mass model to generate the dynamics at each of the nodes in the model. That can be done at various levels. I'll talk about some of the flavors of mean field models in a moment. That gives you the dynamics of the model. You can also stimulate these nodes, for example, simulating deep brain stimulation or TMS or TDCS. You can include that as part of the model itself, and then generate output activity. The model itself generates local field potentials, but using forward solutions, you can also generate EEG, MEG, stereotaxic EEG, which is quite useful for epilepsy research, and functional MRI data, which you can then map back to the empirical data, modifying the model parameters until you get an optimal fit between the model and the data you're trying to model. This is sort of the obligatory equation for the virtual brain. This is a generic equation that sort of captures the main flavors of what happens in the model. In the top, you have the equation highlighted in blue. We've got a change in activity in a population of cells that are based on the current activity of that system, the interactions between that node and the adjacent nodes in the system, the effects of long-range input in the system. Long-range input here includes time delays, which is important in sculpting the brain dynamics. We have both local and global connections. The global connections have time delays. I over here is the input to the system. That's where you would include stimulation, as I mentioned earlier. And finally, we have a noise term. That's basically the noise within that particular node. That can be Gaussian, that can be non-Gaussian, additive or multiplicative noise, whatever you want to do there. The same equation is represented sort of graphically here. You've got a sagittal section of the brain coming through this way, fronts here, backs here. The fibers that are connecting different parts of the brain are highlighted in green and red. What we have over on the right side of the slide is the local model at a particular system. So we have a collection of excitatory and inhibitory neurons, their mutual effects, and then their connections with the local areas that are next to them, the adjacent, basically short-range connections between those cell populations. And that's sort of contained in a chunk of tissue. And then you also have the long-range connections here. And again, those are estimated using diffusion tensor imaging to allow you to estimate the time delays that are going to be there because of the distance between different parts of the brain. So this is a generic equation that captures the overall structure for virtual brain. The details of the population activity really depend on the neural mass, or what's called mean field model that you're using, which I'll talk about next. So in the platform itself, I think there are now about 14 different mean field or neural mass models that are in the program, allowing you to select different kinds of phenomenological or biophysical features that you want to try and model in the system. Because the virtual brain is a network-based model, large-scale network-based model, we don't have cellular models of individual synaptic activity. So, for example, the classic is the Hodgkin-Huxley model. It's the base of a lot of neural net modeling that's out there now. We don't have that in TVB or virtual brain. We do have, though, our different kinds of models that emulate the biophysical properties that generate local field potentials, for example. The Wang-Wang-Deco model, for example, has been quite heavily used recently to look at modeling intrinsic activity in resting state. We have firing rate models. Wilson-Cowan is a classical model that generates firing rates at local fields. And then finally, phenomenological models that are just there to generate phenomenological data. So you can have oscillators that just generate sort of oscillatory phenomena. Hirimoto oscillators is a bit more of a complicated oscillator model. And finally, the epileptor, which we've used to model epileptic seizures and their propagations, which I'll talk about a little bit later in the talk. So these are the sort of range of dynamical models that you can use then to model the individual nodes, which are meant to capture a large number of neurons that are embedded in the system, but then interacting through the long-range connections. And all these models can be described by a nonlinear system of differential equations. This is a brief movie that kind of gives you an idea of the scales. This is a movie created by Petra Ritter and colleagues in Berlin. This is a single subject. You can start by taking that subject and virtualizing it, creating a scalp topography. The person's cortical surface and is triangularized to give it a mesh, essentially. Those can be then converted to regions of interest to identify the different areas in the brain. And then the centers of those areas are shown in the boxes. Using the diffusion tensor imaging, you can estimate structural connectivity between the different nodes. You can also put on an EEG cap on this model and then generate your simulated activity. So the first activity we're generating here is EEG. What you see there is the simulated electrical activity going across the person's head at a relatively fast time scale. We can also simulate, using exactly the same core model, functional MRI data. You'll see that the waxing and waning of activity across the cortical surface is somewhat slower because fMRI bold doesn't reflect blood flow primarily, which is a slower phenomenon. And then finally, the model itself generates source model activity, which has a very fast time scale. The important thing here is that we've used exactly the same core model to generate the three different kinds of activity, which makes it quite a powerful platform to be able to look at both the underlying biophysical features, but how that then predicts the empirical data that you're interested in looking at. So quite a nice feature for the things we're talking about today. You just saw that. I don't want to see that again. Okay, I'll just quickly mention there's also recent developments for using TVB in looking at modeling the mouse. We derived data from the Allen Brain Institute to generate the core structural connectivity, and then are able to do the same kind of simulation platform with the mouse that we're able to do with humans. And more recently, Kelly Shannon in my lab has led the effort to create the virtual brain macaque. So there we have, again, a cortical surface and diffusion tensor imaging to create sort of a canonical model for a macaque monkey. This is just a simulation from that canonical model, but you could then use it again to model individual data you might have from studying macaques. Okay, so that's the first section, just going through, again, the theory, the software platform itself, the core contents of the software platform, and the different features of that. What I'll do now is go through the examples of resting state activity, intrinsic activity, stroke and Alzheimer's disease, and then epilepsy, and then we'll have time for questions after that. Resting state activity, resting state networks, is a small industry right now in neuroscience, particularly in human neuroscience. And it's really focused on the kinds of dynamics you can measure when people are just sitting there not thinking about anything in particular. More formally, it probably should be called intrinsic activity or spontaneous activity. And that's reflecting the notion that the brain, with the exception of when you're in deep sleep or under heavy anesthesia, is always active. And the question is, what does that activity actually reflect in terms of what's happening in the brain? Is it some sort of internal rumination, sort of self-reflection, or is it something else that's really captured in intrinsic activity? The next few slides are basically a compilation of about 6 or 7 papers that where we studied this intrinsic activity in the virtual brain to see what features were necessary to produce these patterns of intrinsic activity, these resting state networks. So the references that were used are on the bottom of the slide. I will mention, I'll try and get these slides up to Figshare, and I'll post the link for that once I'm done today with this presentation. The modeling process that we've used to generate intrinsic or resting state activity has the same sort of flow as I mentioned earlier. We've got diffusion tensor imaging to estimate the connections between different parts of the brain, the regions themselves that are, you drive the MRI and then you do the parcellation, the regional map essentially of different areas of the brain, put it into the computer model, generate the neural activity from that model, and then using a forward model to generate the simulated bold or fMRI signal. From there, you can look at the correlations of the time series, which is also called functional connectivity, and generate that matrix, that's the simulated functional connectivity, and compare that to the empirical functional connectivity. And then we go through a process of fitting to modify the different parameters in the model. So for example, the The strength of the connections in cells, the coupling, if you will, between different parts of the brain can be one parameter you look at. The noise could be another parameter, or some aspect of the local dynamics of the brain itself. I apologize for the blurriness of this slide. This is an example of one of the things we discovered using the virtual brain model to look at intrinsic activity in terms of the capacity of the system to, I'll say, do stuff, more formally, again, process information. So is there something about the intrinsic activity that allows the brain to be more efficient in terms of what it does? And the answer is yes. And what's shown here on the top part is you see this scatter plot with these red dots that are increasing. The y-axis is the correlation between the empirical and simulated functional connectivity, and the x-axis is the coupling strength between the nodes of the network. What you see happening is as you scale the coupling strength, the correlation increases pretty much linearly until it gets to a certain point and then drops off. What you might expect happening, for example, is if you go from a system that's loosely coupled, where you've got individual areas kind of oscillating on their own, as you increase coupling, they become more correlated. You get to a certain point, and then they become a single system and basically oscillate on their own, which means that the system itself will no longer reproduce what you see empirically, and that's why there's a drop-off in the correlation at this point in here. You can also look at, on the bottom is this sort of plane that shows what's called an attractor mapping of the system. This is basically how many different kinds of configurations are there, how many different kinds of networks can be formed in this system. This is a cartoon basically showing that in the far left of this graph here. In that part, you have a very simple system with basically one primary network. As you increase the coupling, the system breaks up into different functional networks, different attractors, if you will. And as you cross this point here, which is called a bifurcation, the system then collapses, and some of those functional networks are no longer available to it. So it's in this point here, what's called the subcritical point, which is very often talked about topic in complex systems, subcriticality, criticality is being important for complex systems. What is it that's important about this part of the system, and where these two lines are put here, point A and point B? We looked at the propagation of activity if you stimulate it in a particular node, in this case primary visual cortex, if you had these different areas in the system, point A and point B. If you stimulate at point A and look at the propagation of the stimulus, you find that the propagation speed is relatively slow. But if you increase the coupling just before this point B, just before the bifurcation, you find that the propagation activity is actually faster. So all we've changed here is basically how tightly coupled the network is. We find that at that subcritical point, the system is better able to propagate activity across the network, suggesting that that point is actually something important for the system's dynamics, the system's capacity to process information. So it may very well be that the intrinsic activity represents what's possible for the brain to do at a particular point in time. What is the number of configurations that the brain can have? How many networks can be occupied? And how does it then impact cognitive function going forward? The one thing you do find, if you do the modeling in a bit more detail, is that these functional connectivity patterns are actually dynamic. So instead of being stationary across, say, a 20-minute period of time, if you look at the patterns of functional connectivity, they will in fact change as you go across segments in time, suggesting that the system is in fact exploring different potential configurations spontaneously as the system moves forward in time. That was shown quite nicely by a paper from Hansen et al., published a few years back, just modeling again the dynamics of functional connectivity. And being able to show that this does in fact represent an exploration. If you look at how the networks are interacting at a given point in time and how that switches, you can map these switches in terms of what's called a manifold, which is shown over here on the bottom left. That these are patterns of interacting networks that form tight clusters at one point in time and then spontaneously switch to another pattern of functional interactions, another point in time, and then go back. This is reproduced both in the, I got it unfortunately covered here, I think this is the simulated data on this column here, and this is actually the empirical data here, and these are the different alpha and beta states. The main point being here is that, again, using the virtual brain, we're able to reproduce these dynamics, showing that the brain is in fact exploring its repertoire spontaneously, and this is what the intrinsic activity of the brain is for, to explore what is possible. There have been groups that have been looking at this in more detail now. There was a very nice paper from Mac Shine in Australia with my colleague Mike Breakspear and others listed on this paper here, just showing this exploratory capacity across different cognitive functions, again showing you this manifold essentially architecture over here. Really nice paper. I highly recommend you go grab it. I think it's open access, which makes it nice to get. This exploration changes as one ages. The way that the systems are configured, how you explore your repertoire, does seem to change as one gets older. And this is some work done by Invictor's group, Damien Battaglia, looking at the changes in functional connectivity, again, the dynamics of functional connectivity in aging. And these are actually real data from subjects of different ages, and you can see that as you go from the 18 to the 30 to 55 to 72, the switches are actually reduced. Basically, the fact that these matrices are more red suggests that there's less switching of these networks as one gets older. That was also shown in a recent paper from Joana Cabral in Spain. These are actually real data as well, just showing that the patterns of these switches and how they switch is actually predictive of cognitive status in healthy elderly, which is a nice way of linking then the dynamics of the internal state of the brain, intrinsic activity in aging, and how that then relates to cognitive status. So a nice sort of collection altogether. Okay, that's a fast tour of the intrinsic activity. Again, I'll mention that these slides will be available on Figshare. I'll post a link later on through Twitter, at least. The take-home message from that first section was basically focusing on the fact that we can, in fact, model the intrinsic dynamics of the brain, and that these moment-to-moment dynamics relate to the actual and potential cognitive dynamics that are there for all of us. Let's now switch to the example from dementia. The example comes from a single paper actually published by my former student, Joelle Zimmerman, who's now in California, where it's sunny. And she worked with our colleagues again in Sydney. Parminder Shakdev provided us some data on what's called the Sydney Memory and Aging Study. Michael Breakspear and Alistair Perry were very important to help us get the data together and put them into the particular model. It's a largish data set, not massive, but large-ish, with 73 controls, 35 people with amnestic MCI, and 16 with AD. There is a follow-up study which we're going to use to replicate these data, but this is sort of the first proof of principle that we could get something useful from modeling dementia in terms of predicting cognitive status. Model details, we again used MRI to get both the geometry as well as the connections between the person's brain using DTI, and also used the bold functional MRI to fit the models. Used the Wang-Deco model to do the dynamics, and we're fitting both local and global parameters in the model. This is a busy slide showing again the workflow for the model. It's similar to what I showed earlier. Again, we have diffusion tensor imaging to do the structural connectivity. We have resting state fMRI, which generates our empirical functional connectivity. We have our model itself, which is based on each individual, so this is now a personalized model. So we use their structural data to construct the structural basis. We use the computational model to impart dynamics for the node and generate simulated functional connectivity, which is mapped back to the empirical functional connectivity to optimize the parameters to improve model fit. And then we use the parameters themselves to map back onto the cognitive behavior of the entire sample. So one thing that's a characteristic of Alzheimer's disease is there is a progression. It usually starts with what's called the limbic system, which is a part of the brain that's quite important for memory. Includes the hippocampus, entorhinal cortex, parahippocampal gyrus, and other regions. And the question we had there is, can we look at how the limbic subnetwork changes its interactions with the rest of the network in dementia, and use that as a way to see how well we're able to predict cognitive status. So basically, the embeddedness of the limbic subnetwork may change with the progression of dementia, whether that embeddedness can be used to differentiate the different clinical severity. What's shown here are just the basically descriptive statistics for the raw data. We've got the structural connectivity on the top in the blue bars, the functional connectivity in the sort of oranges peachy bars on the averages between the groups. You see, as you might expect, that patients who have Alzheimer's disease show reduced structural and functional connectivity in the limbic subnetwork, less so for structural connectivity, but the functional connectivity is in fact still reduced in the Alzheimer's group. So we'll create three models essentially. One is the full model, one is the limbic subnetwork, and then we'll look at the comparison as we transition to an embedded network to see how that works out. Again, a busy slide. The main point here really is that if you look at this embedded network, all of the networks do predict cognitive status, but the embeddedness does a slightly better job of predicting the cognitive function, attention, language, executive functions, visual memory, semantic memory, as well as verbal fluency across the population. The bar graphs here show on the top row the different parameters in the model. We have the global coupling, and then the local parameters, which are the inhibitory excitatory interactions within the local cell populations. So all of them are actually predictive of the cognitive status on the other side. The embedded network does slightly better in terms of predicting the cognitive status of the individual, the cognitive function of the individual. So that's cool. The question that comes out of this, though, is this modeling effort, all this effort to go through and getting all these different details, does it really provide anything more than what we have if we just stuck with the original structural connectivity or functional connectivity? So we compared that and looked at how much better the model itself did in predicting cognition compared to looking at just structural connectivity or functional connectivity, mapping that to cognition. What you see here is a plot of basically the normalized covariance between the model parameters and the cognitive function. The model itself is in this red distribution. These are bootstrapped estimates, so that's why there's a distribution across. And then the structural and functional connectivity in green and blue. And it should be quite obvious that there's quite a huge improvement in terms of the predictive capacity when you use the model to predict cognitive status. So actually the model does better than using the empirical data by itself. This might seem somewhat paradoxical because the model is in fact based on the empirical data, but our supposition is that what's happening here is the model actually then distills from the data that part of the data that's most important for producing the biophysical parameters and eliminating all the other measurement noise that's inherent in things like MRI, where you get a lot of noisy signal and you have no way of parsing signal from noise. The model helps essentially by boosting the signal and eliminating some of the noise of the measurement. Okay, so the biophysical parameters may have some clinical utility. The supposition, we actually haven't completely tested this yet, but it may in fact provide us with a mechanism to assess both risk and decline. We can extend that a little bit more by looking at stroke models, and specifically models of stroke recovery. So this is an example of a patient who's had a stroke. You can see on the T1 weighted image, which focuses primarily on gray matter, you can see that there's a hole here in the left hemisphere, left parietal cortex, which is indicative of a stroke. On the T2 image, which emphasizes the white matter differentiations primarily, you see again a similar mapping of the stroke here. The question is, if we take patients who've had these strokes, look at their model, can we use the model parameters then to predict how well they're going to respond to physiotherapy, in terms of motor recovery from the stroke. In this particular model, we're focusing both on the balance of global coupling, which again is this general long-range input scaled by the diffusion tensor connections themselves, as well as a local excitatory and inhibitory balance. We're looking at both, again, local and global parameters and how that balance changes in the face of stroke, but also how that we can use that then to predict recovery. This is an example of what's called a parameter sweep. What you see here are some global parameters. We have the conduction speed, so how fast signals are propagating along the connections in the model. And we have the coupling parameters, which are basically how tightly coupled the different connections are. And what we're doing is we're scaling these parameters across a broad range and then mapping how much variance there is in the network. And that's what the heat map shows here, is as you scale the parameters, where does the system start showing high variance, which is indicative of an active system, a complex system. So it's a sort of a surrogate marker for how well we are producing a reasonable model. So you see that the speed itself doesn't make much difference across the y-axis, but the x-axis, as you increase coupling, you have a sweet spot here where the global variance is maximum, suggesting that that's an optimal critical point for that particular system. This particular model is based on one of the healthy controls. We can do that for another healthy control over here. You see that the regions of transition are roughly the same between these two controls here. We compare that to three different stroke subjects and find that some stroke subjects show a similar, at least a qualitatively similar topology, whereas others show quite a different topology compared to controls. There's about 40 subjects in this sample and an equal number of controls, but these are just sort of snapshots of two examples in terms of the global parameters in the model. We can also model local parameters. Here we're looking at the changing of the excitatory and inhibitory balance in the local nodes. Inhibitory is modeled as GABA coupling, excitatory is NMDA coupling, and again we're modeling global variance in the system. And you see as we scale the parameters, again this is a healthy control, there's a sweet spot where the variance is in fact at maximum, suggesting that that is the critical point for this person's network. We do the same thing here, mapping the parameter space in a couple of healthy controls here and here, indicating that the sweet spot is roughly the same for the two healthy controls, and that's in fact true for most of the healthy controls. Some stroke subjects are similar to healthy controls, others are quite different, and you see again some examples down here. We can plot or correlate the model parameters obtained from each individual with clinical metrics that indicate how well they're responding to therapy. There's a thing called the Fugl-Meyer, therapeutic index that's used for motor recovery. And here on the left, you see the scatter plot between the excitatory coupling across patients and how well they respond to therapy immediately after, early on in therapy, essentially. You see the people who have less excitation are actually doing better. If you look at them further on, this is about a 1-year follow-up, I believe, you find again that those people who had higher excitatory coupling, those that did worse essentially. So it's such that hyperexcitability in the model is actually predictive of worse recovery of function in physiotherapy. So this is a relatively small sample size for clinical studies, but suggesting that the personalization in TVB can be used to predict recovery of function, suggesting how you might in fact then guide therapies for individuals based on what you see in their individualized virtual brain models. So both the dementia as well as the stroke model just suggest that there's a change in local global balance, and that balance is in fact indicative of how well people are going to recover from particular diseases. In the stroke model, it's a bit more local, which you might expect because there are focal changes, but there also are some global coupling changes. In things like dementia, where you have sort of broader network changes, that balance of local and global function is probably going to be more critical in terms of understanding what happens as the disease progresses. So the last example is on the virtual epileptic patient. This is work again led by Victor Yersa in Aix-Marseille University. The model itself was built to capture the phenomenology of epileptic seizures. So it wasn't so much focused on trying to model the individual biophysical features that are important for generating seizures, but rather generating time series that would be consistent with what you'd see if there was a seizure. So the phenomenology of focus was basically shown here, and this is actually a single unit recording from a rat hippocampus done in the colleagues lab in Marseille, Christophe Bernard, who works closely with Victor. What we see here is as the system approaches seizure, it starts having fast discharges, and then goes to a quiescent resting state, ramps up again, shows seizure activity, goes back to quiescent state essentially, and goes back and forth between these resting states and these fast discharges. So that can be modeled as a shifting in what are called fast and slow variables that are inherent, again, in systems that have multi-scale architecture. We think about multi-scale architecture in terms of spatial architecture. For example, the brain has different kinds of spatial distributions. You have cells within populations, within areas. Each of those also have different time scales, faster and slower, and the fast and slow systems will actually influence one another as the system goes through its behavior. So you can represent this spiking behavior in terms of the interaction between two processes, a fast spiking process shown here in the blue lines, and then a slow process that basically enables these faster discharges. So the slow process changes gradually. You have what's called an exit bifurcation where the system goes back to rest. The slow variable increases slightly. At some point, the threshold is reached and it allows the system to start spiking again, oscillates back down, the system stops, and so on and so forth as it goes back and forth between rest and spiking behavior. Represent that in a phase flow diagram, which is kind of nice to see the trajectory of these behaviors. You have the variation in the slow variable, oscillating back slowly, and then it has an entry bifurcation where you get the fast discharges spiraling up. Exit bifurcation, the system again moves very slowly, and then back up through the system again. So this alternation of fast and slow discharges allows applications of maths that characterize multi-scale architecture, specifically fast and slow systems. And that is called the epilepter, that particular model that does the spiking behavior that oscillates between fast and slow variables is called the epilepter. That's what that says here. And the question is whether or not this phenomenological model can help to model the propagation of seizures in patients. So this is an example case study. There are many more now. There's a paper that Yersa published in Neuroimage that shows a number of case studies. This is one of them, a female patient who has bilateral temporal epilepsy, also has a malformation in the hypothalamus that has hyperconnectivity between the two sides of the brain through the thalamic connections. You see here is the angiogram from the patient on the top. These are locations of what are called needle electrodes for the stereotactic EEG. This is the location of the patient themselves, and this is a CT scan showing you the location of the needle electrodes. Each of the points indicates a contact where you can measure activity along the axis of the electrode. This next slide is the data from that particular patient. Each of the colors indicate the location of an individual electrode. So the top is a brown color. If you look up on the brain itself, you see this brown electrode with a large ball indicating high activity, which is where the seizure focus, one of the seizure focuses are. The other electrodes you see through here, the contralateral electrodes in frontal lobes and in the contralateral hippocampal and temporal lobe areas. So these were modeled. The nice thing about modeling these seizures is the clinical data themselves are kind of restricted in the sense that they have to only have measures from where they put electrodes. In the model, you can generate activity anywhere, so it basically can both model where there were data measured empirically, but also where there were not data. And that may help you get a better idea of how the seizure might be propagating. And that was the task, essentially, of doing a virtual brain model with these data in this patient, but also subsequent patients, which I'll talk about in a moment. And so here, this is the actual virtual brain model for this patient. We basically scaled different epileptogenicity values to look at how the system propagates a seizure going from the original focus in the right hippocampus to the contralateral area. So the seizure onset here, indicated by red arrows. The seizure spreads into the thalamus, which wasn't actually measured in the original clinical data. You don't see any traces here from thalamus because there's no electrode here, but you do see a lot of activity in thalamus here in the model itself. And it's likely because of this malformation that's in the thalamus that could be helping to propagate the seizure between the two hemispheres. The seizure then propagates to the left hippocampus into the temporal lobe and left hippocampal gyrus, and then terminates there. So using the model, they're now able to get a better idea of how the seizure is propagating, and then that allows them, the surgeons, to know how they might intervene to isolate the focus and do the neurosurgery to isolate the system so it doesn't actually propagate further. So this is generally a broader workflow, just showing what the goal is, and also showing how well then the virtual brain does in predicting the seizure focus plus the propagation of the seizure compared to what the clinicians predicted from the clinical data. So again, the goal here in the middle is to predict what is the epileptogenic zone, where is the seizure starting from, and then identifying where the propagation zones. Trying to isolate the epileptogenic zone is basically the key when you're dealing with drug-resistant epilepsy. We're trying to help prevent seizures from moving forward. And what you see here is basically the data fitting on the slide C, where the excitabilities are. The highest likelihood for fit was actually in the right hippocampus. We mapped that back to what the clinicians predicted, and there was a fairly good agreement, although some clinicians actually thought of the other hippocampus was the seizure focus. These are some additional fit metrics, which I won't talk about now, but you can go to the original paper from Victor Yersa and his colleagues, published in 2016. The key thing is how well does the model then help in terms of predicting the clinical outcome from this particular, using a particular model. And I'll just focus on the Engel score here, which is basically a metric to say how well did the person recover from the seizure. Are they seizure-free, or at least the seizure is reduced? And a 1 indicates good outcome, a 3 indicates a bad outcome. And the higher the discrepancy was between what the model predicted, what the clinicians predicted, the worse the outcome was for the patient following surgery. So that outcome was actually used as the motivation then to go and launch, to validate basically launching a larger clinical trial, which is now underway in France, involving about 400 patients that they're using virtual brain to help provide additional support for clinical decisions made in terms of how to intervene with the patient. So it's quite exciting now, because actually now have a case where we've gone from a computational model that's based entirely on computational theory to now being able to use it to guide clinical practice potentially. So again, a very exciting opportunity for the virtual brain now to move into helping clinical decision support. Before somebody asks, I'll make it very clear though that we're not saying that the only answer that is the right answer is coming from TVB. Of course, like most clinical decision support tools, the idea is to use that as one piece of evidence to help guide how the surgeon will then treat the patient. So it's an additional piece of information that the clinician can use to guide their decisions on how to treat the patient. Okay, I'm just going to wrap up now quickly with a couple of points. Talking about, generally speaking now, what are the good parts and bad parts in terms of the virtual brain platform and how we've set it up. The first one is that it's model agnostic. By that I mean that the particular way you're modeling the dynamics of the system is a choice that you can make. we do assume that you're going to be modeling large-scale networks, but how you do that in terms of whether you use oscillators, whether you use the epileptor, whether you use the Wilson-Cowan equations, that's entirely your choice. So we allow you that choice to make decisions on how you're going to model the data. That gets back to our original discussion saying that if you're going to model a complex system, you have to make decisions on kind of what you're going to focus on. What is the phenomena you want to try and reproduce, and how you're going to do that in a particular model. The other advantage is that it does allow you to integrate empirical data directly, allowing you to make what's called essentially a personalized model, which is quite a powerful tool. Also scalable, so you can in fact do very coarse models where you've only got a few regions of interest, say 64 areas, but you can do what are called surface-based models, which allow you to go up to several hundreds of thousands of different kinds of nodes. So the spatial scale is, in fact, a modifiable parameter in the model itself. It is easy to integrate in an empirical workflow. In my lab, for example, we're using it routinely to try and link with clinical data in the case of Alzheimer's disease, but also as a complement to the general cognitive neuroscience studies to be able to link what we measure empirically back to what we measure in terms of the behavior of the individuals. And that's a nice way to integrate the modeling framework into a traditional experimental workflow. And as part of the workflow, it allows you to make inferences about the biophysical parameters that are underlying what you see empirically. It's basically a family of generative models that generates the data based on underlying assumptions about the processes that are important for the model. Not everything is rosy, of course. There are bad things. It's a nonlinear system, so model fitting and identifiability is a formidable challenge. You can get deceived, if you will, in terms of getting a model that seems to fit well, but it's actually sitting in what's called a local minima. So you have to be very careful on making sure you do a fairly intelligent parameter search to try and come up with a model that's the most consistent with the data as much as you possibly can. In its current version, the virtual brain has no plasticity, so we can't model learning in the traditional sense or neurotransmitter distributions. This is a solved problem. The next generation, probably the next release, will have those in there, but currently it does not have neuromodulatory effects or plasticity in the general sense. I did mention that the decision we have made in terms of the virtual brain is that we're modeling large-scale activity. So the inferences that we're making are based on the population level, not on the synaptic level. And that's, again, a decision that we made in terms of how to do the model, where to focus the model itself. And finally, of course, like any nonlinear model, any complicated model, the interpretability is often a problem. So it becomes a question of how to link up what you see in terms of these complicated nonlinear operations, the oscillations of local and global functions, and trying to relate that back to the cognitive phenomena you're interested in looking at. That's a good challenge to have, though, I would say. So it forces you to think a bit more about the transition between the emergence of the dynamics at the brain level and the concomitant emergence of the cognitive functions at the brain level, at the cognitive level, I should say. So the emergence of brain, emergence of behavior, and how to link those two. And that's, in fact, what I think the virtual brain is good for looking at the interaction between dynamical systems. So what I'll do now is I'll stop here. I'll leave this slide up, which is basically the take-home messages, and I'll turn the microphone over to my host to, I guess, adjudicate the questions. Thank you.
Great, thanks, Randy. That was a phenomenal talk. That's a lot of great information, and wow, what a cool project. So thank you for presenting to us. I'll go ahead and open the floor to questions. If you'd like to ask a question, you can either put it in the, I'll monitor the chat window and try to ask questions that come in through the chat window, or you can unmute yourself and ask a question. So we do have one question from Mateo to ask you, can you use, may you use template MRI? Maybe Mateo can unmute and maybe add some exposition to that. No? Okay, well, we can come back to that. Is there anybody else who would like to have a question?
I think I know what he means. I can maybe ask that.
Oh, okay, go ahead then. Yeah, please.
Yeah, I think what he's talking about is probably the parcellation schemes. For those of you who are immersed in MRI research in humans in particular, there's a huge, again, another industry on parcellations. And we can, in fact, accommodate pretty much any parcellation that is being used currently. A lot of that's basically, it's incorporated in the workflow. So if you can specify your parcellation, that can be easily integrated into the workflow for virtual brain.
Great. So I see that Lorette is unmuted. I'm guessing that Lorette has a couple questions that she'd like to ask. Lorette, is that true?
Yes. Can you hear me?
Yes.
Yeah.
Okay, I have just one. First, congratulations, Randy, for very, very interesting work. In fact, I have two questions. One, when you talk about the personalization of the profile that you are extracting from the model that can guide interaction, reaction to various interventions, to what extent are you also taking into account the dynamics of effects of those interventions in relation to individual differences at the patient level into structural and functional dynamics there? That's my first question.
Okay.
And the second one will be this multi-scale processing that you are very brilliantly putting together. And currently you are remaining multi-scale within the brain. And it's a bit related to my previous question. To what extent could we progressively have different layers of multi-scale influence, whether it is the intervention that you customize as a function of individual dynamics or other aspects of multi-scale phenomenon?
Two great questions. And it's almost as if I prompted the question, because the second one, which I'll come back to in a second, was a paper we just published, or actually put up on bioRxiv, I should say, talking about a framework that does exactly what you're talking about, where you allow it to look at multi-scale interactions between things like brain and behavior. The framework doesn't depend on particular operations, so it can be used for other scales as well. But let me come back to that in a minute. The first question was about if we include the individual differences in dynamics in the models. And in fact, yes, that's the fitting features themselves. So we have the structural information that provides sort of the template, if you will, or the pipelines, if you will, for the model. But the parameters themselves are derived based on fitting the dynamics of the individual subjects. Whether you talk about normal cognition or clinical cases, we're trying to fit the individual models to what you see empirically. Particularly important in stroke recorders, because you want to make sure that you're capturing the seizure patterns in the patients to best estimate what the propagations patterns are. So yeah, it's a big part of virtual brain, is using the individual person's dynamics to actually fit those models directly. In the multi-scale component, Victor Virsa and myself have been working on basically a framework called structured flows on manifolds. And that's really embedded in dynamical systems theory. It's really focusing on how to link up nonlinear systems across different spatial and temporal scales. And the notion there is, again, because it is a generalized framework, it's equally applicable to brain as it is to behavior, as it is to genetics, as it is to molecules and other complex systems. So that paper was just uploaded on bioRxiv, and I'm happy to send a link for that too if people are interested in looking at it for me.
Oh, thank you so much. It seems so timely, Sean. You will have an assignment to study this closely. Thank you. I will leave space for others for questions.
Thank you.
That will be only one of the 15 assignments that I have been given to study Randy's work. All sorts of aspects, but no, I think it's great because in reference to many of the things that we do here at McGill, we're not really dealing with dynamics, and that's I think is a critical component of looking at systems. So that's why I really appreciate the Virtual Brain Project. So we have a couple more questions from the chat window. There's a question from Ron Reisenbach, which is a little bit loaded. If you had $1 million, what improvements would you spend it on?
Get a better PR firm. No. I think right now the main bottleneck in a lot of what we're doing is processing speed and increasing the usability and the time it takes to do the simulations. The example from epilepsy is actually a good example of what we have to do to take the virtual brain and move it into clinical use. For the use in the clinics, you have to basically freeze the software development. So you can't go in there and tweak the code when you're in the middle of a clinical trial. So to make it usable, you have to optimize the code so that it's accessible by the clinicians, but also runs very fast. And that allows you to then to put through a lot of data very quickly to do the kinds of validation you need to do for other clinical indications. Epilepsy is underway in that domain. We're not there yet with dementia or stroke, but I think that would be where I would focus a lot of the investment, is making sure that the code is optimized for the clinical expression and runs very fast, so it's actually useful then in the clinical domain.
So a bit pursuant to that, follow up on that. I mean, obviously, if you're simulating the dynamics trajectory in the brain, there's only so much you can do to speed that up in terms of parallelization, because you have to. I mean, it's time-dependent simulation. Then,
yeah,
you can't do time. You can't do time 100 before you do time 4.
Right.
Have you considered any sort of ensemble approaches that might allow you to parallelize at some level the time domain?
A little bit. This is something I've been talking about with Victor in the context, and Petra for that matter, in the context of what's happening in the collaboration between Virtual Brain and the Human Brain Project. Mm-hmm. Because there we're looking at trying to increase the spatial and temporal scales of the model, and it gets to the point where it's just not feasible to do a simulation in one's lifetime. Mm-hmm. So the notion there is trying to find an effective way to parallelize it. Maybe even using sort of hard-coded systems like the Nest computer, for example, in Jülich. But we haven't gotten very far in that, but that's definitely in the workflow.
Yeah, because I come from in a previous life I had worked a lot on molecular dynamics, and the protein folding problem is a very similar type of issue, although with a random event that occurs as opposed to so you have to run it's even probably a bigger, longer time scale problem. But then folding at home does like sort of an ensemble approach with like Markov chaining together this trajectory to kind of circumvent that. Yeah. So I thought there might be some wisdom in that kind of approach that might help you there.
I'll definitely look it up. Thank you.
Cool. Okay, so we have a follow-up question from Mateo in the chat room. How much is the parcellation scheme important for the good fitting between simulation and empirical networks?
Okay. I don't know the direct answer to that. We haven't tested it directly. I do know that when we change parcellation schemes, the fits per se don't change too much in terms of the qualitative metrics. What will change is how predictive the biophysical parameters are of other behaviors. And that's basically just because you're changing spatial resolution. So I don't actually know what the optimal parcellation size is. And that's something that we obviously have to look at in great detail if we're going to do clinical derivations. But for a given parcellation scheme, you can get decent fits, and you can get decent fits from the same data set using different parcellation schemes. It's actually more a question of the details of the biophysical phenomena you're looking at. You might lose if you smear over too large of an area. That's been basically our experience so far.
Okay, great. Are there any other questions from the audience? You feel free to. Okay, yes. So we have one from Eugene. Hi, Randy. Can you please clarify again what kind of empirical data can be simulated? You mentioned resting state fMRI. I am assuming we can also use resting state EEG. What about event-related potentials data or frequency tagging type EEG paradigms?
Yeah, thank you for that question. Yes, all the data I mentioned, EEG, MEG, intracranial EEG, LFPs, functional MRI are currently simulatable. In terms of things like ERPs or frequency tagging, can be done. It's not that it can't be done. It just hasn't been done. They're more to do with how to actually do that in terms of the input parameters that you would use to modify, for example, the evoked potential in a certain part of the brain. So there are ways that the stimulation parameter in the model is that I term I showed in the general equation. That's one entry point that can be used to model things like stimulus input and propagating that across the system. There is a paper that was published by Misha Scherner et al. In eLife. I think it was early last year, pardon me. It's again from Petra's Ritter's group, where we used EEG to drive the local dynamics, source modeled EEG to drive the local dynamics in the model to generate the ensuing fMRI data. And that's a way to actually fuse two different data sets, but you can also think of using a similar kind of approach to model cognitive paradigms where you have a stimulus coming in, for example, in V1 or auditory cortex, and looking at the propagation patterns as a function of modifying stimulus intensity and so on. Hasn't been done, but doesn't mean it can't be done.
Okay, great. Any other questions from the audience?
Laurette has one more, but
okay, please, Laurette.
I will give priority to others.
No, go ahead, please.
Okay. So I was looking yesterday, Randy, to the diversity of your work, and I was extremely impressed by so many things. I have in front of me here a paper that where essentially you and a long list of collaborators work on the famous Walter Mischel study where delay impulsivity or delay discounting was measured at 4 years old, and then you get them back at 40 years later, and then you get higher dimensional neural network for the low delayer. If you and you have done also in parallel extensive work on change in brain structure and dynamics with aging. If you were to kind of not build upon this kind of cross-sectional observation of where they are 40 years after, and ask to kind of what would be your vision of how this structure and dynamics will have evolved over time, over aging, in those who started their life low or high delayers. And I think that this is really our group, the Brain to Society, is really to say we need to bring the understanding of the brain to understand real-world behavior and life course and so on. So I'm just curious to pick your brain on that.
Wow.
If they had not told you, I'm not an easy lady.
No, it's like you've also done your homework too, which is kind of frightening. So that's actually this is a great question and a great segue, and I think so. Part of the work that we did in measuring or modeling the intrinsic activity of the brain was done to see how that mapped onto functional capacity. And so there was a parallel set of empirical studies that we did, again, cross-sectional data, unfortunately, looking at measuring what's called multiscale entropy, which is basically a measure of, you can think about information processing or predictability of signals across different time scales. And it's basically, to be sort of colloquial, it's kind of a way of saying how well does the brain process information? Is it processing it well or not well? And what we found in going from kids to adults to old adults is that these entropy measures increased into adulthood, and then there was a time scale dependent shift in aging that happened. And that time scale dependent shift was both in terms of whether you got fast or slow activity, but also spatially. What ended up happening was that the brain seemed to shrink and focus more on local dynamics, but that was actually predictive of good cognitive function in healthy aging, which changes sort of how you think about what happens as how the brain works, that there is this sort of continued balance of local and global dynamics. Early in life, it's important to have global dynamics that try to integrate information as well as it possibly can. But it might be the case that as we go forward in life, we want to have more segregation, which allows us to have more options to choose from. Higher entropy, more choice, for example, more capacity to delay gratification, for instance. So I'm going to go out on a limb and say that what we could probably try and think about doing now with something like the virtual brain is to look at how this interplay of structure and function move through life, how that relates to the broad spectrum of cognitive functions that the humans are capable of, and see if there's a way to link them up more formally. There are data out there that can do it cross-sectionally. It's hard to do this lifespan study, of course, but the data do exist. So we'll be starting actually on Friday, tomorrow, doing TVB baby, and trying to get some data that allow us to look at these landscapes of brain dynamics in babies and in kids, and see if that landscape then can be used to see how it supports the emergence of cognitive function, how it supports the complexity of cognitive function, and how that landscape changes then as we get older to maintain cognitive function, but also is it predictive of risk of cognitive dysfunction going forward. And that's really where I'm going to focus a lot of my work on, actually, and that's getting back to these structure flows and manifolds that I mentioned that Victor and I are working on, is using that then to link back from the dynamics of the brain to the dynamics of cognition.
Very, very interesting. If I can push a bit further, in this Walter Mischel 2 data point, and you did the looking at the structure and function 40 years after. Yeah. But I'm just wondering whether there was most likely low delayer than they were 4 years old that have grown up to be longer term, longer delayer. Whether you could kind of observe a different dynamics at 40 years, whether there had been this shift from i.e. impulsivity or whatever we call them to learning to have more control over time vis-a-vis those who started their life low delayer and will end up low delayer. So I'm just wondering if you were having the empirical basis to explore this.
I don't. I mean, there might be data out there that have that kind of capacity to look at the change in things like impulsivity and control going forward. We could do that in patient populations, but we have to actually have also the data prior to the onset of the disease. But I don't have that available to me, and that's something that, again, the framework we're putting in place will hopefully be able to accommodate both sort of low-level cognitive functions and sensations, but also I'm hopeful that it will also allow us to look at these more higher-level functions like morality, cognition, and so on, and trying to do some relationships in that way. It provides the quantitative link to these phenomena, and I think that's actually what's missing right now. I'm hoping we're able to do that with things like virtual brain.
Thank you so much. Back to you, Sean.
Sure, yeah. I think that's, we have time for one more question if somebody has one. No? Okay. Oh, go ahead.
Sorry, it's Eugenie again.
No worries.
In the stroke model,
Go ahead.
That
Go
was.
ahead.
Hi, can you hear me?
Yeah, we can hear you now. Go ahead. Sorry about that.
Okay. Just going back. So the local parameter that's estimated, is it the case that it's a local parameter for across all the spatial locations in the brain, so independently of where there's stroke? So you don't specify anywhere in the model where the stroke actually is, or you do?
In that particular model, no. All we do eliminate the nodes and the connections from the stroke area, but the parameters themselves are global for the entire model. So every node has the same parameter for local excitation inhibition. That is being changed now, but even doing that, we're able to do a fairly good job in predicting clinical outcomes. I'm hoping we'll do better with allowing individual nodes to change, but right now we didn't do that for that particular model.
Great. Well, okay. Well, I think that's all the time we have for today. Again, thank you, Dr. McIntosh, for giving our webinar. That was a wonderful talk and good questions from the floor. We'll be having our next bridge webinar at a to-be-determined date. Randy will find this funny. It'll be Bratislav Misik, who will be speaking from McGill University, who will be talking about his work on network modeling in the brain. So it should be complementary and similar. But, yep. So we'll look forward. We'll send out some information on that, and when we get it scheduled down, and we'll hope to see everyone there. And thank you again, Randy. It was a really great talk.
Thank you. Take care, everybody.
Thank you, Randy.
Bye-bye.
Bye.